Thank you. Thank you for the introduction. So I'm Yasuyo Minagawa from Keio University. You can see my name, my Japanese name here, Minagawa Yasuyo. So you know some meaning from these characters. And I'm from Keio University first. And I have to say that, first of all, thank you for this invitation to such a great symposium. And I'm very honored to be here. Thank you. So I'm continuing. So first, thank you. So first I will introduce my affiliation, Keio University. Keio University is in Tokyo and the Kanagawa area. And Keio University is the oldest private university in Japan. But the history is shallow, it's just 160 years. But Yukichi Fukuzawa is a founder of Keio University. You can find his face on the Japanese bill, the most expensive one, because he's famous for leading liberal democracy in Japan in old days. Okay, so now, and then I direct Keio Baby Lab in Tokyo, and we are interested in diverse pattern of development, focusing on language and social skills. That's why we perform a longitudinal cohort study with TL and EL infants. Today's talk is about this cohort study. So here I will explain what TL, EL infants stand. TL infants means infants at risk, at typical likelihood of ASD. They are just typically developing infants. On the other hand, EL infants is infants at elevated likelihood of ASD, autism spectrum disorders. They are sibling infants of ASD children, brother and sister. Because ASD has a genetic factor, so they have a high risk for being a ASD symptom. But we know only less than 30% of sibling babies has a ASD symptom, but rest of the 70% of babies also have some kind of developmental delay sometimes. Yeah. So I will explain about this longitudinal study in more detail here. And this is, so we follow behavior and brain function longitudinally from 3 months old to 3 years, 1,000 days. And then we measure their brain function at the age of 0 year, 3, 6, 9, 12. And also we measure their behavioral types of development. And also we apply developmental testing. And because ASD cannot be diagnosed until 3 years, at least in Japan, maybe later. So we don't know if these babies has really ASD symptom in the future. But we describe their patterns of brain responses to language and social stimuli here for 2 groups of infants. And then we examine the correlation between these patterns and later language and social development, so that we can predict which brain or neural factors can predict ASD symptom. And actually, some infants show a problem at some stages. And then in that case, we have a collaboration of intervention. So, so Professor Yamamoto's lab apply intervention for such infants. And this is how we test babies. We behavioral testing, eye camera, and we measure motor development. And also, today's talk is chiefly about FNIRS study, functional near-infrared spectroscopy. This uses light to measure the blood changes inside the brain. So this is very handy method to apply to infants. And this is the mobile type of FNIRS device. All right, so today's talk consists of 4 topics. They are all about TL and EL infants, except for the last one. But the first, I will show mother-infant behavioral interaction at 6 months and later development. And the three topics, rest of the three topics related to neuroimaging study. The second one, brain response to the mother's speech and stranger's speech in TL and EL infants. Third one, hyperscanning mother-infant brains during parenting in TL and EL infants. The final one is multimodal hyperscanning of mother-child active social interaction. Okay, so now the first topic is about mother-infant interaction at 6 months old and later development. In this study, we measure parent-infant social interaction. I mean, we video recorded their interaction using a paradigm called still phase paradigm. I will explain the paradigm. The paradigm consists of 3 phases, as you see here. The first one is free play. Mother and infant play freely without toys, and then for 3 minutes. And then we ask mothers not to respond to babies' behavior at all. So even though infants give a smile, mother has to keep their face still. So that's very difficult for 2 minutes. And then the final stage, mother returns to normal mother. So she can have, this is a free play period again. And we had 25 and 23 EL and TL infants, but we now have doubled number of infants, but the result doesn't change at all. So I will show the data. And based on this kind of video data, we applied a behavioral coding of video, such as mothers and infants' vocalization, smile, eye gaze, and touch, and contingency. This is important because we have some results about this contingency. Contingency defined here is mother's immediate response to infant's social signal, like smiling and vocalization. So that immediate response is marked as contingency. And we also applied some questionnaires and behavioral testing in the course of follow-up study. Now I show you some results. These are mother's social signals, such as smile, touch, contingent response, depending on the phase, play and still phase. These are frequency of these social signals. And red one indicates EL infant, and green one indicates TL infants. So maybe you can see a difference between the phase. So, of course, during still phase, mother is told not to respond. So the smile and the touch and still, contingent response is smaller than the pre-play period. But one thing I have to point out is there was no difference between EL and the TL infants group. So no difference between red and green. And we also applied the same analysis to infants' behavioral data, and we didn't find any difference between the groups for any type of social signals in infants. But we found a difference in terms of the relationship between maternal contingent response and infant fasting during the still phase period. So as you see here, only the group TL group, green one, had a correlation, positive correlation between contingent response and fasting. And fasting during still phase reflects good attachment because infants know usual mother is not always like that. So if they have a good attachment, they change their behavior during the still phase. Then, so this means the more frequent the caregiver's contingent response, the better attachment infant has. But on the other hand, as for EL infants, we didn't see such tendency, the red one. So it seems like maternal contingency has no effect on fasting or attachment for EL infant, elevated likelihood of ASD. And also we found interesting relationship between mother's contingent response and subsequent later language and social development at 9 months and 12 months. The more frequency of mother's contingent response, the better the development of language and social ability for 9 and 12. Only for TL infants, the green one. EL infants, this one may have a kind of opposite tendency anyway. But only for TL infants, better parental contingent response enhance the infant's language and social development. Also, this continues to up to 2 years old and also 3 years old. And the better the frequency of mother's contingent response, the better the expressive vocabulary in this case. The last one was data from behavioral testing, but this is the language questionnaire at 18 months and 24 months, only for green one, TL infants. Only for the TL infants, the more frequent, yeah, that's the same. So, and there was no correlations for receptive vocabulary, only in this case, only for expressive vocabulary. Okay, so now we know mother and infant interaction is important for language and social development. Now, I will go back to that point later, but now I move on to the neuroimaging study. First one, the second topic is brain response to the mother's and stranger's speech in TL and EL infants. So we also tested EL and TL infants. And we presented them mother's speech and stranger's speech. It's just infant-directed speech spoken by mothers and strangers. We presented them many times for 15 seconds. And we measured their brain activity from frontal and temporal areas using FNILS. And we also analyzed the brain activation as well as brain connectivity, which means the connection between one brain and the different region brain. Before I show you the 6-month-old data, I show you neonates data using exactly the same paradigm. This data is interesting. So this is the brain connectivity in response to mother's speech. So this is left side. On the left side, you have a language network, language area. In front, what we call IFG, and that's anterior language area. And there is a posterior language area somewhere here. That's include STG. So we see, I think this is the first evidence showing neonates language activations network. This was only observed in response to mother's speech, but not for the stranger's speech. And another interesting result is the connectivity from right STG, which corresponds to right auditory area, to the frontal area. So I guess on the right auditory area, neonates can identify the vocal characteristics. So this is familiar or not familiar. And then that connects to prefrontal cortex, including OFC. And maybe OFC is a reward area, so mother's sound may be reward worthy for them. Okay, now I show you the recent result of 6-month-old infant for TL infant and EL infant in response to mother's speech and stranger's speech. So as you see here, TL infants show a large and also many activities on the brains, not unlike EL infant. And so the place circled in red is left STG area, and the pink one is orbitofrontal area, which I just mentioned, which is related to reward processing. And so Yeah, so only in TL infants, maternal speech activates voice processing area for both hemisphere and attachment-related area. OFC is also famous for to be related to attachment, but not for EL infant. And if we directly compare between TL and EL infants, so we can still see OFC activities and both side of auditory area. And furthermore, we found a significant difference in activation of IFG, the anterior language area. So TL infants showed larger brain activation involved in speech processing and attachment than TL infants. But interestingly, there was no difference in response to stranger speech. Okay, and we also analyzed this kind of how this activation is related to later language development. And we found something. So the oxyhemoglobin means the activation, brain activation. So the more, larger the brain activation, more greater the expressive vocabulary at 18 months old, only for TL infants again. No significant relation for EL infants. Okay, now I move on to the results for connectivity, the brain network from right SDG area, right auditory area. Here again, TL infants show the connection between our right auditory area to orbitofrontal cortex. And also, but not for the stranger's speech and not for EL infants. They didn't show significant connections. And this one is the connection from left SDG, which also corresponds to posterior language area. And interestingly, this EL infant showed the connection between posterior language area and anterior language area again, only for mother's speech and not for stranger's speech. And EL infants showed some significant connection, but not between IFG and SDG. So in response to mother's speech, TL infants showed strong brain connectivity between IFG and SDG. Language is a language network. And we also performed a correlation analysis between that's IFG and SDG connectivity and later language development. And this is, language score at 9 months. Only for TL infants again, the strengths of connectivity between IFG, SDG, and IFG, and also OFC, orbitofrontal cortex, had a positive correlation with later language understanding score. And this was not observed for EL infant. So why are there less activation and connectivity for the mother's voice in EL infants? Maybe social stimuli, including voice, are not a reward for EL infants. And there was a previous report. Actually, we found this paper after we got these results. But adults with ASD have weak functional brain connectivity between voice-specific region, which is auditory area, and the reward system. So only so this is the different point. So SD, this adult SD does not have a good connection with voice area and OFC, these areas. So the familiar voice of their in infant case, the familiar voice of their mothers is worth the reward for the TL 6 months old infants. That's why they have a connection with OFC. So connectivity from our SDG to OFC may serve as a kind of biomarker to predict SD. Maybe they are not preference of familiar voice. So maybe it's one option to measure this brain connectivity. Now I move on to now I move on to the third topic, which is hyperscanning mother-infant brains during parenting in TL and EL infants. Hyperscanning is a simultaneous measurement of brain imaging. So usually we have a brain system called social network, as you see here. For instance, mentalizing network or mirror neuron system. So previous studies found social-related brain regions. So the brains of two people may create a social brain network. It's been reported from previous study, only for adult study. So create a social brain network together by synchronizing between them. So even young infants may show brain coupling with adults. So maybe mothers and infants' brain also may show a synchronous movement activities. And also there may be differences in mother-infant brain coupling between TL and EL infants, even at an early developmental stage. So that's why we performed a hyperscanning study with mother and infants. Participants were 43 TL and 17 EL infants. In Japan, it's very hard to recruit SD sibling. Anyway, so we had 3 conditions. So first condition is breastfeeding. And also the second condition was holding rest condition, where mother held her baby and stayed still. And the final kind of control condition is a separation rest condition, where experimenter, it was actually me who held the baby, so I held the baby, and mother stayed in the same room, but she didn't see her baby. And we measured their brain at that time. And the next slide is about the analysis method, but I don't go into detail. Just we measure the brain activities from 2 persons, mother and infant. Then we examine the coherence between them for particular frequency band. Okay, so now these are results for interbrain coupling. And first, I will explain this main effect of condition. As I told you, we have 3 conditions. But we found a stronger brain synchrony for breastfeeding than control and holding. Actually, I expected some synchronization even for the holding condition, mother held the baby. But we didn't find significant synchrony at that condition. But it's very strong synchrony for breastfeeding, as you see here. So this is mother's. So these are FNIRS channels. We placed the probe here and measure the brain activities from these areas, mother and infant. So this is too much. So we to understand clearly, we just picked up top 15 strongest synchrony from between mother's brain and infant brain. Like this. So these brain areas are synchronizing. And so as you see here, from some particular brain region like TPJ, temporal parietal junction from infant brain, that is part of the social brain network. Has many significant projection, not projection, but synchrony between these mothers and infant brains. And also in case of mothers, the OFC also played a role here. So now we examine the correlation between these synchronization and later development. For instance, the synchronization from OFC had a positive correlation, this one, positive correlation with crying duration during still phase. So as I told you, this is an index of attachment. So this means the more synchronization, the more the attachment is. Also, it has positive correlation with positive vocalization during free play. In case of this TPJ, synchrony with between mother and the infant. There is a positive correlation with parental bonding score and the mother's sensitivity. this means the stronger the parental bonding, stronger this synchrony. And we also, I didn't show you the data here, but we also find a positive correlation between this one, the synchrony, and later language development and social development as well. Okay, so now this is the final study, but this is still going on. Just I will show a very brief result. This is multimodal hyperscanning of mother-child active social interaction. Because in this infant study, this was our first challenge for hyperscanning. So we wanted to reduce the motion because it damages the data. So, but we know how to how to process those kind of motion artifacts. So now we challenged hyperscanning during mother-child active social interaction. Now, this time we used a mobile type of F-News, as you see here, and we measured facial electromyography, the activity of facial muscle. And also we applied AI-based pose estimation to extract their behavior. Because this is still going on, I will just show one result. But before that, I can show you how it was going on. So this was the mother-infant child interaction. So they are playing a kind of pretend play of hospital, doctor and patient maybe. So she has a thermometer. And maybe she takes, injection. Toy. Yeah, this is our hyperscanning. So okay, so now I will just show one slide. So we extracted the behavior of mutual gaze, and this is the synchronization during their mutual gaze. So this is mother's brain, and this is child brain. We found the strong synchrony between medial prefrontal cortex, which is also part of social brain, and also it's connected with OFC area, and also MTG and OFC. Okay, so now, I'm running out of time, but so this is a summary. And we've been seeing a neurodiversity of mother and infant interaction. Mother-infant as for the behavioral study, we found only for TL infants, maternal contingency impacted on development. But I don't think for EL infants, maternal contingency does not work. So I think it seems like contingency given by EL mothers sometimes not adequate. They are sometimes too intrusive and too much. And also, our intervention project team also use contingency for our intervention to ASD kids. So it can be used. And for instance, we use it as a contingent imitation to induce vocalization from nonverbal children. So, but I think we were discussing yesterday, but we need an adequate type of contingency. For instance, we may have to enhance a kind of contingent social signal. Okay, and we found a different pattern of connectivities in response to maternal voice. And so, and this kind of TL infants connectivity led to better language development. And we found a similar tendency for between I think, yeah, I forgot to tell this, but yeah, I forgot. But we didn't find any group difference for synchrony. Yeah, so I just put the data together. So we didn't find any difference between TL and EL infants. They both showed a synchronization at their age of 3 months old. And also, okay, so, and this also led to better language social development. And I have to point out that, so every 3 neuroimaging study, we found involvement of orbitofrontal cortex. So I think the reward-related system is a fundamental neural base for communication development through parent-child interaction. So I will summarize it using this kind of figure image. So I will show the different developmental patterns between TL and EL infants. So for instance, so if baby gives a smile, kind of social signal, so it is a reward for mother. So her OFC activates. Then she will give back the contingent response by speech or smiling. And then that is also a reward for infants. So that's kind of interaction between them enhance child's interest in familiar voice and face. And also that's increased attachment. Also, infants got more interest in human voice and mother's voice. So that's enhance the language development. So they may construct a language network between IFG and SDG. And that's also lead to better language and social development like this. But in case of EL infants genetically may have a weak interest in human. So, but when the baby smiles to mother, then So maybe, of course, the mother's OFC will activate, and she will give back contingent response. But as I told you, their contingent response may sometimes not adequate. So, and also, infant is not interested in such a human signal, so their OFC may not activate so much. So maybe not much interest in voice and face. And that may lead to weak language and social development. And I have to say that orbitofrontal cortex, which is related to reward network, is a critical factor to enhance language and social development. And that drives language and social development. And to enhance that kind of OFC, maybe you need a suitable or efficient type, adequate type of contingent response and adequate mother or parent and child interaction. So that's all my study. Thank you so much.